more distant from nutrition. It's still about plant nutrition, but it's about yield stability. Getting the same yield in all environments, different environments, maybe in different years. And one of the questions that I myself ask to my colleagues and also to myself frequently is, do we really need a lot of physiological knowledge? Do we need to know all the ammonium transporters or phosphate transporters to have really good growing plants or optimal plants? And do we need this for abiotic stress tolerance? For stress tolerance, because we are always exposed to variable environments, and I don't know the situation exactly will be in Costa Rica, but it's expected that in Germany at least, with climate change in the next few decades, we will have not only warmer climate, but also more extremes. More extremes means long terms without rain, then followed by very heavy rains, and all weather extremes, hot, dry weather, together with a lot of sunshine, followed by humid and rainy seasons also, that will be more challenging for plants. And in this context, we are also very interested in whether plants remember previous environmental conditions, whether, well, they are not only responsive to some conditions, but they really may remember even over many seasons or generations, transgenerationally remember some previous environments, at least the molecular features in the plant have been identified that this should be, or could be the case, but actually we don't have much evidence for that. So we'll talk about this a little bit today, and I want to start with a figure, how normally myself, agronomists, breeders also, that also work in the field a little bit, or only in the field, how they look on yield stability, how they look at the plants and investigate yield stability. What do? The researchers, they look on plants in real environments, real fields, the environmental conditions that we have for plants in the environment that we are breeding for. So we have maybe wind, sunshine, rain, heavy light, many different high temperature or low temperature, cold and spring. All this is exposed to plants grown in real environment, which means that typically most of our crops we grow in monocultures currently. So when we think about maize, we have a big maize field and plants are neighbored by other maize plants. They are exposed to insects, to other biotic stress, and it's more or less unpredictable. We agronomists or plant breeders or also plant nutritionists, we then grow these plants under best practice, and after the growing season we harvest and look at the final harvest, the final yield, and that's the critical point: how much did we get over all these stresses, how much residual yield is still there, despite we had all these stresses and the plant responded somehow with some tolerance mechanism or so to these stresses. That we look at these plants in the field, in not too small areas, where each plant that we is really surrounded by other plants, this immediately it's clear that it has some benefits when we analyze it in the field, for example for drought tolerance. When the plants have neighbors, they have shading from their neighbors. This shading, one part can be good, and the other part, with too much light, is good. With not enough light, it's bad. We have overlapping root zones. So the plants acquire the nutrients not only from soil, but in competition with their neighbors. Under realistic conditions, with maize for example, the roots very seldom meet each other. And we have not too many overlapping extraction zones for nutrients. And it depends, of course, on the nutrient. When we are considering nitrogen and nitrate, then the nitrogen is coming from relatively far away with the mass flow of water towards the plant, and then we may have overlapping extraction zones for nitrate and nitrogen. For phosphorus, this is not so much a problem. Very seldom the roots meet, and the area that is explored by the root to extract phosphorus from the soil is typically relatively small. But for a drought condition, for example, this also is important because some plants, well, they meet the water extraction zone of the neighboring plant, or the next neighbor even, and this has several consequences. First of all, one consequence is that this plant here takes away the water from its neighbor, and it may be beneficial even if this plant here saves a little bit of the water, that the neighbors also have sufficient water under dry conditions. Under normal evolutionary conditions, we don't have these monocultures. We may have individual plants, and the plant will win that gets the most of the water and takes away the water of all the competitors. So cooperativity in these kind of scenes may be really a relevant thing, especially for water uptake and water usage. In these settings, we normally, myself, I'm not so familiar with these settings because my setting is more this here. We have a small greenhouse. Our stress is very well designed. What we do is we have a control condition in the greenhouse, and when we say, now we want to get the plant to have a dry condition, or we want to have the plant to be exposed to nutrient deficiency, we directly do it at that time point. So the time point is exposed by us, not by the weather outside, and it's not so unpredictable, and we compare the control versus stress condition. This control versus stress condition we can't compare in the field because we don't have the control. We only have exposure to all the abiotic stress. And that's more my field where I'm coming from, plant molecular biology. Well, and yield stability, surprisingly, is in my community not so much a concept. The concept in molecular biology and molecular genetics is more stress tolerance. That's what we talk about. Yeah? It's already different names. It's plasticity, the plant responds in some way to some negative environmental condition. Or we also talk about robustness. Yeah? And during my recent work in Hohenheim, let's say in the last decade almost, I learned that in my community, in this agronomy faculty, many different researchers or other professors always have a different view on what is yield stability or abiotic stress tolerance. And I want to share a little bit of these ideas that I have to discuss with you today. So, what is yield stability? Many of you probably are agronomists and have a very good knowledge about what yield stability is. My naive expectation of yield stability is something like this. We have a certain plant exposed to 5 environments, maybe 4 different soils, and one environment maybe is just a repetition in the next year with different weather conditions. It's also a different somehow environment. And then in total we may have 5 environments for this crop, for this one genotype, and we can compare it with other genotypes. And I would say, well, this has similar yield across environments, that's yield stable. And now the question is, who agrees with me on that and who disagrees? So first, who agrees that this is yield stable? Okay, only a few. What do the others think?
This is maybe for the current concentrations of CO2, for example, and it could change in the future if you change the...
Yeah, okay, it's not optimal maybe for all future conditions, yes. My point, this is a slightly different point that is very important for the breeders always, but also for agronomists. My point is that a different genotype, that is for example like this here, would be selected by the breeder. It may not really show the same, let's say, yield stability, but we are much closer to the optimal yields. And that's one basic feature of this yield stability. When we talk about it, we don't only want to have the same small yield in all environments. We want to have it close to the genetic potential of the crop that we are thinking about. For example, for winter wheat in Germany, we would have something like typically 9 tons per hectare. And here, this setting here, this genotype, would be much better than genotype number 1. Although we have one condition where apparently the yield was not very good. Okay. So very important to this terminology of yield stability is that we are close to the genetic potential. And I think, yeah, this makes really a lot of sense. Why? Because if we consider a very small, tiny plant, probably it will always make this small yield in all conditions because it's so small. It grows very slow, very cautious in this environment, and well, it's not growing at optimal rates, and it's not giving us the optimal yield. So to be very close to the yield potential, that is one fundamental basis of this concept of yield stability. Okay. Yeah, and now as a plant nutritionist, I have to say, well, how do we keep it with the nutrition if we don't supply nitrogen, for example? We may get this here. It may sound very yield stable, but it's at very low level again. So should we consider different fertilization levels? High fertilization level, low fertilization. Is this also yield stability? What do you think? I see some people shake their heads. I also would say, well, I would be happy to have maybe different management practices, but I would not include bad fertilizer practice. I would say, well, everything should be at least more or less close to optimal management and close to optimal fertilization, as far as we can tell. That to me has more sense to be considered as classified for yield stability. With poor nutrition, if we lack a certain nutrient also, we anyhow will get not super duper rocket science, I would say. Okay, so then it comes to the question, how many environments do we need? And I sometimes talk with my statistics professor colleague in Hohenheim, and he tells me for really good analysis of genotypes in German environments, he suggests about 40 environments. 40 environments is much too expensive for any breeder, for every company. So 40 environments is really on top what the scientist wants. What is practical is probably something like 3, 4 environments. More or less diverse environments. Yeah? And that's typically what breeding journals also require from their authors, that they have repeated their experiments in, let's say, at least 3 environments. That's frequently asked for. Okay, so that's the agronomy side of this yield stability. And we in Hohenheim, we are relatively strong in that side in Germany, and because we are relatively strong with that, we also have some molecular biologists working in the agronomy department, but we have stronger groups even at the University of Tübingen, which is close to Hohenheim, Stuttgart, and the Max Planck Institute there. And therefore we recently applied for a project where we look at yield stability in ever-changing environments at the Ministry of Sciences in Germany, and we got funded and got some money for it. This we investigate in a so-called Regional Research Alliance on Yield Stability. We are still in progress, and I don't show you much data today of this project, but for me it's very important that we have that, because that's something completely missing yet, and I will go a little bit more into detail why I have shown you already a little bit why I think we use different language and different experiments to show more or less similar things. But we have then also, we are asking different questions, and that should be unified somehow. Okay. So, again, for the molecular part researcher, well, here he is, thinking about Arabidopsis. You have heard also research from Arabidopsis yesterday from my laboratory. So that's what we like to talk about, because we have mutants, we have small plants growing fast, growing in the greenhouse, well controlled. We have all the tools for molecular analysis, that's very good. But actually we have a shitty plant that is just a weed in reality, and it's not even a weed in the field, it's a weed growing in the northern hemisphere, very often close to small streets, also not so much related to weed in agronomy. What we look at there are really molecular processes. We identify individual genes, we characterize them, we show their functions, have gene families, we have very nice genetics, we have a very small genome, all this is fine, we can sequence a lot. Most of it we do in controlled environments. Sometimes we go outside. We, for example, also, last year we tried to do a field experiment with Arabidopsis. It was a disaster because they did not germinate as quick as we wanted, as normally in the laboratory. So in the end, the student had to work on barley, which was also grown in the same field. Yeah, and here we have the breeder, looking not at individual plants here, but looking at communities of plants. Sometimes we have also some competitors here, but in maize fields typically it's controlled by the farmer, that we have strict monoculture always. It's real weather, I would say. We don't have the real control condition as we have here. And we have to do it also with different traits. Different traits means here we have a single gene, we are interested in this gene, it looks like an ammonium transporter, oh, let's check it in all ways whether ammonium is important for this gene. And here we look at a trait of, well, stress resistance against different stresses at the same time, biotic, abiotic, whatever. Okay. The breeders are very often happy to have the plant as a black box and not knowing all the gene regulatory networks that are there. They want to have, well, high yield compared to low yield. They have different also tools to bring the plant somehow to be a little bit bigger. Okay. So some questions that we ask, for example, did we lose or did we gain components of yield stability during the breeding process? Currently it looks as if we gained yield stability by selecting always the good genotypes. Yeah, so at the moment, I think breeding can say yes, we probably are good, also for yield stability. But I fear, I showed you in the first day or the second day some data where we looked at growth of maize on low P environments. And I showed you some data related whether modern genotypes of maize are still mycorrhized, similar as old ones. And one conclusion was yes, they are still mycorrhized, but they seem to have a smaller root, a trend for smaller root in modern genotypes, which may be related to our fertilization scheme. Okay, so when we look in the field, or generally look at the physiology of plants, we have several abiotic stress resistance scenarios, and they depend on the local environment, and typically for all stresses, we have these kind of three major categories of how the plant can react with the stress. Similar with cold, for example, similar with freezing, similar with, what else can I think of? Well, let's look for it with drought. We can have the escape strategy. What is the escape strategy? It is that we escape the very hot times of the year. The plant tries to grow in those times when it's not yet too hot, and then it tries to grow as fast as possible, which means maximal gas exchange, maximal losses of water, and maximal income or uptake of CO2. Stomates always open. And then finish the growth before the really dry season begins. When there's still sufficient water, we grow maximum, maximize gas exchange, stomata open, early flowering. And early flowering means early seed set, early ripening, early mature seeds. That is an escape strategy to be finished before the terminal drought begins. Of course, only applicable to an environment where we have this kind of terminal drought. Okay, escape strategy. The opposite, basically, is more or less here in this avoidance and tolerance mechanisms, which means that we do exactly the opposite concerning the stomates. We close the stomates not to lose too much water. So avoid loss of water. Avoid loss of water by closing stomates, by deep rooting to assess further water. And then maybe the metabolism does not have to adjust for the lack of water, because we get more water from the roots and we do not evaporate too much water via the leaves. So the metabolism can work at the same, more or less, water content, and we avoid that we have problems with not enough water. That's one of the very lazy, simple strategies. And a little bit more complicated is, of course, to be really tolerant. Then we have to adjust the metabolism, the morphology, everything, to really assess the little bit less of water and work with less water in the metabolism. We have osmoprotectants like proline, beta-alanine or other osmoprotectants. GABA also. Or have even completely different metabolism like the C4 metabolism, the crassulacean acid metabolism. Okay. To have a look a little more detail in drought, I want to cite some data from Prof. who is a researcher in France, working on maize since a long time on drought. And he has very nice concepts and data published to that. And I just want to summarize some of his ideas, or the community of the ideas, for different drought scenarios, with different plant strategies to cope with this type of drought stress. So one adaptation to drought stress might be the growth maintenance and high stomatal conductance under water deficit. Is positive under relatively mild water stress or dry stress, but negative in severe terminal droughts, of course. Seed abortion can be positive because it can allow proper seed filling for some seeds at least, not for all, under severe stresses, but it's definitely negative under mild stress, because then the seeds are aborted and we have very low yields. Root growth, root system architecture is a trait that plant nutritionists, of course, are very interested in, because at the same time when we take up more water, we may also take up more nutrients. Maybe a positive trait for some conditions, but not always. Yeah. Sometimes the bigger root is also negative. Avoidance in general of this loss of water, cell protection, yeah. Several genes, a lot of genes are known for this kind of trait, from molecular physiology, can be sometimes good. Increase water use efficiency, a positive trait in very dry environments. And what do we really want? Do we want the genotypes with high yield in most conditions, or genotypes with specific tolerance to water deficit? Well, when we want to consider yield stability, we want it to have high yield in most conditions, probably. But we may have a certain field, a certain area, where we know the predicted weather will be always more or less the same, and then we may want to have some specific tolerance to water deficit. Okay. Yeah, I give you also a table from François for these different stresses here, and the variable for phenotyping, you can have a look. Abundance of known genes, yes, very often you have a lot of genes, sometimes you don't have many genes for a certain stress. You see the positive, the drawback, the scenario with maximum positive effect, and the scenario for maximum negative effect. When François discusses these things in his paper, he basically comes to the conclusion that any trait or trait-related allele can confer drought tolerance. You, as a researcher, have simply to design the right drought scenario. And that is, when you think about this in more detail, you feel that this may be really right, and I strongly believe that this is right. And when you look at different papers, especially by plant molecular biologists working on different drought scenarios, we very often design our drought scenario in a way that we get these positive effects of mutants. But this does not help, in general, to improve drought tolerance, but it shows only that depending on the condition that you use, you get a certain drought tolerant phenotype or allele. I want to show you the last part of this table, also especially for this year, the root architecture, yeah, where he says, well, there's not so much known. Well, there is actually, there's in rice one gene and allele, the locus known, the drawn one, locus, and it was associated with better water use efficiency, but again, we have to consider these water use efficiencies and drought scenarios, especially in the field, and not only in rhizotrons. More boxes filled with soil, with a plexiglass cover where you can visualize the root. Where we have deeper roots in the draw one, or in the draw one allele is important for deeper roots and more water uptake. Sometimes when we invest more into deeper roots, we have less investment into roots at the surface. Sometimes we have less nutrient uptake than there. Deeper water gets available, but lower nutrient availability, especially with phosphorus in upper layers. Okay. And that reflects a little bit this balance that the plant roots have to do with water and phosphorus that I showed several times in my slide.
I have a question.
Yes?
Where you say the trait it says contain vegetative growth, and you're using NTDI and proxy detection for variable for phenotyping. What is proxy detection?
Proxy detection. Oh, that's a good question. I did not see that. I did not look at this. What is proxy detection? I'm sorry. I can't tell you. Does anybody know? Yeah, there's some damages, probably. Yeah.
I understand you can get like alcoholic chemistry using NTDI, but I've never heard of proxy detection.
Yeah. I really missed that. I did not look at this. Overlooked it. It also says this number 4. I should have a look what number 4 is. Okay. This citation. Sorry. Okay. Okay. So my conclusion from this kind of table, and especially the tolerance to drought, is, and when we asked this question that I had here, one of the first questions on my first slide, how much physiological knowledge is needed for abiotic stress tolerance? Well, for specific droughts, it's good to have these physiological knowledge, because then you can directly see and evaluate whether this specific drought occurs here. But in general, the fast-growing crops with really high yields, they can't be tolerant to everything. Yeah? They are specialists in some way, specialists to certain environments or certain environmental conditions. And the best is, you know all the typical stresses. They are more or less constant over the years. You have repeating stresses also, and not too many changes, with, for example, with climate change. You know that you have to do it with mild drought, or with severe drought, or with terminal drought, with early drought, or repeated drought, or whatever. Then physiological knowledge seems to be efficient. Otherwise, I think we should forget about physiology. We should just select the best ones in many environments. Yeah, and then even optimized plants for a certain environment may be sensitive to other types of drought. And I think most clear example is when we have these escape strategies compared to real tolerant plants. Escape means the plants grow in those times when there's still sufficient water, at maximal rate, stomates open, a lot of gas exchange via the stomates, at highest rates of photosynthesis, excessive water loss, but they terminate their growth by inducing flowering, inducing senescence, filling of the seeds relatively early, when the terminal drought begins. And then really the opposite we need for plants in mild drought environments over the whole year, not terminal drought, but dry conditions over the year. Then we always have to reduce storm-mantle gas exchange during the autumn. Okay. We can have experts, highly expert genotypes for targeted environments, certain environments that we know are special in a way that this genotype may really cope with, very nice. Little evidence that single individual genes help in these oligogenic, multigenic traits. And I think breeding companies, also universities, will make a cost-benefit analysis. Which environment is good enough to really breed a specialist for? We very often want to have generalists. Also breeding companies, that they can get a lot of money for these generalists. So, from this, I will come back to this in a few minutes and show you just some genome sizes, what the plants, the crops show us. And what we know is that the crops typically have really large genomes compared to some native plants like Arabidopsis. Arabidopsis here is at the very end. It has 5 chromosomes, 5 haploid chromosomes. The haploid set is 5. There are some tetraploids, but most Arabidopsis are diploid. And the haploid size of the genome is relatively small. It's 120 mega base pairs, million base pairs. Okay. Here are some crops I added, also banana. You're probably much better expert. I hope this is correct. It has a relatively small genome, similar to some trees in Germany, fast-growing trees like poplar. It's also similar genome, a little bit smaller even. What are interesting genomes? Maize, rice, soybean, major crops of the world. In maize we have 10 chromosomes. Chromosomes do not have anything to do with the haploid size of the genome. We always have, for all these crops and native plants, we have between something like 25,000 and 40,000 expressed genes in these crops. That's typically the number of expressed genes. Always the same number of genes, but chromosomes are different and genome size is completely different. Maize is especially problematic because it has this very, very large genome. Red wheat, only recently fully sequenced, is a nightmare in terms of repetitive sequences and genome size. But even that was recently fully sequenced. Durum wheat, spaghetti wheat, I call it also. Slightly smaller genome, still pretty big. Related to that, the barley, relatively small genome. What do I want to say with this? I want to say with this, we have more or less the same number of genes, but very, very different genome sizes. It means that, first of all, the genes are probably covering more area in the chromosomes, and the spaces between the genes and the space between the exons, the expressed regions of the genes, between the genes, these regions are much larger in crop genomes like maize or like wheat. So the spaces between the genes and potential regulatory sequences there are much bigger. Okay. So now we come to something, first lesson of genetics. Well, you're all familiar with that. Mendelian genetics. And we learned that at school. Mendelian genetics And I would suggest, although we learned this at school, or I learned this at school, most of you, I guess, also learned it at school, I want to say, well, it's not exactly correct. You will be surprised, well, Mendelian genetics, wow, this is common knowledge that must be correct. I would say, well, genetics seems to be somehow different in crops. And I will try to explain why I think this is not entirely giving us the correct picture. So, what is Mendelian genetics? Mendel, he recognized that there is some segregation of individual genes, alleles, from parents to filial generations. There is an independency of inheritance, so individual alleles are independently segregated. And there are dominant recessive traits, one gene, one trait. Very convincing data, and he got these very convincing data with a crop, with pea, and he had yellow peas, and he had green peas. He had these two parents, one yellow, one derived from a yellow pea, and one other parent from a green pea. He crossed these, didn't matter which was the father, which was the mother, and he got a uniform F1 generation, and this generation he selfed, and then he got this 3-to-1 ratio. He had a dominant trait of yellow peas and a recessive trait of green peas in the F2 generation. And this is somehow a normal crossing breeding scheme that we all know more or less, and we believe, yes, this is how genetics is more or less working. Yeah, and I would suggest this works for very few genes, in fact, only for this green-yellow qualitative tree, where you really see this very big difference, you see yellow or green color of the peas. That a single locus, however, determines the phenotype is true in only very, very rare cases in crops. That you have a single gene determining really the phenotype in that way. That's extremely rare. This is one rare example where, well, apparently genetics started, green and yellow pea, but in reality, many times, several genes contribute to a trait, or several genes contribute to a phenotype. I think today we still have some of these traits. I don't work personally with these disease resistance traits. Sometimes they have one gene, one phenotype, susceptibility to a pathogen or not. And I want to try to explain you why I believe that Mendel was only right for very few cases, and nowadays genetics should be thought in a different way. But this is the difference between qualitative, green-yellow trait, or quantitative traits, a large smear of different phenotypes. So let's consider a monogenic qualitative trait, green-yellow color of peas. One gene is responsible for it, found in one of the chromosomes and one locus, and when you investigate the phenotypes in a number of lines, you get a very nice result. You get either green or yellow color of the peas, and or you get resistance, tolerance, or whatever. You have sensitive, non-sensitive plants, or whatever. The phenotypes are one or the other. Two possibilities. That's the simplest case. We have it very rare. Oligogenic traits, we would say nutrient uptake. We need several nutrient transporters or family to take up a certain nutrient, and then we need certain other transporters for transferring it to the shoot or so. We have several genes that are important there, and the phenotypes that we have in a population of plants is, well, found in several categories. With intermediate ones, we have phenotypes with lower uptake, with more uptake of the nutrients. And what we normally have, and the trait that is most important, that yield is more or less underlying, is a multigenic trait. Nutrient efficiency or yield are these kind of traits. Causal are many regions, probably not only these ones, probably many more. Maybe most regions in the genome are responsible for yield or for nutrient efficiency. And the distribution of phenotypes then of a population of plants is then the Gaussian distribution. Well, this was the Gaussian distribution that I could draw on my computer. So it was irregular, but it should look like a Gaussian distribution. Yeah? That's what you normally get. So what you get is not this black and white or green and yellow situation of phenotypes in a crop and in crop genotypes in a population of plants. You always get normally this here. A Gaussian distribution of phenotypes, to which many genes, or all genes, and the background depends. So one example, I just picked this up because I thought, well, you have much more knowledge about the banana and problems with banana growth, but I heard that this tropical race 4 is really one of the big problems now coming to Costa Rica. And I guess these kind of disease susceptibilities, they are very often, not always, I don't know exactly for tropical race 4, but these are very often monogenic or traits that have involved only very few genes. And there, I would say, yes, biotechnology may be a possibility to make plants disease resistant. Transgenics in that way, however, are probably not useful to make plants tolerant or resistant to abiotic stress, because when you look at this here, how many transgenic events you need to make really the distribution, this Gaussian distribution of phenotypes, more shifted to the value that you want. So in this kind, one gene, one phenotype difference. With disease resistance, we can imagine that transgenics may be useful. For most of the traits that I was talking about this week, nutrient use efficiency, phosphorus use efficiency, nitrogen use efficiency, yield, transgenics are probably a secondary option for these abiotic stress tolerance. I mean, you know, monogenic traits, as for example the sensitivity to a herbicide or so, this is used with biotechnology, very successful in many crops. Again, monogenic trait. Okay. So, I want more or less to partly finish this part here. Biomass yield nutrient efficiency and abiotic stress tolerance always are quantitative traits, which means we have this smear of phenotypes, more or less Gaussian distribution of phenotypes. And it means that many or even all genes and alleles contribute to a phenotype, but all these genes contributed only part of the total phenotype, only a small amount. All have very small effects. And quantitative continuous traits are then analyzed by quantitative genetics. That's mostly statistics, what our readers do. And very often these traits are not inherited as individual genes. So, not like Mendel tells us. They are inherited as normal distributions, Gaussian distributions, with variance around the mean, which means that the context of the genetic background also also matters. It's not only the individual loci matter, but the context. All the other background genes together, they also matter a lot. And with this, I come to the conclusion that for quantitative traits, many, all alleles contribute, as we just saw. Depends on the genetic background, all small effects. The conclusion from this knowledge is, of course, that small effects are more relevant than these few genes. And then the molecular biologists, like myself, that look always at individual genes, which function do they have in the metabolism, they are not very useful here for making crops more tolerant to abiotic stresses. These few genes are not so relevant. It's more all these small effects. This is not simply Mendelian genetics. I would not say Mendel was completely wrong, but I think we have to understand this better, what this really means, that we only inherit not a phenotype by itself, but a mean with the Gaussian distribution around the mean. That's what we inherit. And I think that's the relevant thing for nutrient use efficiencies. That's why probably we have still very few cases where individual genes were successful to improve nitrogen use efficiency, phosphorus use efficiency. I showed you very few examples, and to my knowledge there are not more that have been really proven to make modern genotypes really more use efficient to nutrients. Yeah, so individual genes very often will not improve phosphorus use efficiency, nitrogen use efficiency, or in general abiotic stress tolerance. So I have to have a look what comes next. Well, with this, I just want to first ask whether there are questions or comments. Yes?
This would mean that breeding would be more in a traditional way?
Yes, unfortunately, I'm really, I find it very unfortunate that my highly advanced molecular methods that I'm so proud of, that these probably will not help us so much to really improve current varieties. Yeah? More traditional statistical analysis and testing in many environments, I think, is the way to go. Yeah? In many cases. In many cases. We can have specific environments where this molecular biology is really used.
When you have, for example, QTL analysis and
Yes.
associated markers
Mm-hmm.
with a small or medium effect on the QTL,
Mm-hmm.
would it be a useful tool for selecting genotypes for early selection?
Mm-hmm.
Of course, it's only a small effect, but if you find a marker associated to a quantitative trait, would it be useful to use in a breeding program?
Mm-hmm. Well, it has been shown that sometimes these QTL analyses are quite successful, but when you look later whether plants with these kind of QTLs back-crossed into current genotypes also, whether they are really much better than the previous plants, typically they are not much better. Maybe they have a small effect. So in the end, this QTL analysis suggests that, let's say, 20% of your variance is depending on this QTL. And you kill this gene and identify the beneficial allele, and you bring it into other genotypes. And typically, what you get is a very, very small improvement, which is not 20% better or something, maybe 1%. Going a little bit along these lines, that it's all small effects. There are effects that are bigger and some that are smaller over the genome, but probably all contribute a little bit. Yes?
I would say that one really good use of QTL in molecular biology would be to identify the sex of the plant. I mean, I can think of papaya, I can think of pimienta. I can think of cannabis,
Mhm.
where you would really want to identify before you let the plant grow to full term, whether it's male, female, or hermaphrodite. I think that's a really good application of molecular biology that you can really look into.
I completely agree, and I will later also show you a few things that probably by this kind of statistical analysis you would not yet understand. You need the molecular biology to better understand certain things like sex, determination, and also we will later talk a little bit about epigenetics and memory. So without molecular tools, you have no idea to get a hand on this. I think there's still enough reason to think that molecular biology can be very helpful. But what I want to say is that our dream that we improve the current crop variety with just adding 2 or 3 of our super genes will not help. When you look, for example, for maize or for all known crops that I know of, we have commercial varieties only for biotics, insect tolerance, herbivore tolerance, herbicide, Roundup Ready tolerance. We have, I showed in my lectures, we have one maize variety that has been made in the US, I think it was DuPont, that made this abiotic stress tolerant variety. It's one variety that they say, oh, it's more stress tolerant. I think it's the only one on the market, you find, surprisingly. So probably when you have the problem of too many stresses, or you want to improve your yield in a certain environment, the farmer would change the crop, probably, before changing to a transgenic variety, or you would change the flowering time of your crop to be resistant against terminal frost, for example. So changing the flowering time for a certain crop is definitely also something that is very valuable. It can increase overall yields. So, yeah, if we don't have further questions, then I suggest we make a coffee break. And after the break, we ask whether our plants can remember previous environmental conditions, if they have stress memory or trying. Effective. come to the second part of the lecture, which will be on plant memory. And my question here is whether plants have the possibility to remember previous environmental conditions and previous stress. And actually there are different names for this kind of memory in plant research. And yeah, we will discuss this a little bit. And of course, I would also like to have your help, whether you have experienced some situations where you think, yes, the plants remember some previous stress. I will first make a few words before we then get into a discussion. So my statement for the beginning would be, well, plants are sessile organisms. They can't run away as we can. So that's how many of my colleagues start their lecture. This means they have to have this adaptive mechanisms to adapt to new environmental conditions, because they can't choose where they live. They have to take what they get. So most likely, I would say, plants should remember very little or not remember the previous stresses, because if otherwise a new stress would occur, they would be only adapted to this one certain stress and could not respond to the others, and they must remain adaptive to environmental changes during all of their lifespan somehow. So I would say, from the beginning on, I would be first skeptical to have this kind of environmental memory. But when we look at the molecular physiological knowledge that we have, we, for example, know that the plants, well, they have a day-night cycle. They are exposed to light during the day. They are exposed to no light during the night. And the plants are very much used to this kind of day-night rhythm. So when you transfer plants that had been grown in a day-night rhythm and you put them to a dark room, what you see is that this periodicity of gene expression during the day still continues a while, a few days. Yeah? So the plants still remember somehow that there was this periodicity of the day. And one argument that the plants are always still responsive to new environments, one argument in favor of that, is that when you look at the genes, metabolic genes, that are expressed during a time cycle during the day, let's say a 12-hour cycle of light followed by 12 hours of night. When you look at gene expression, it has been looked on gene expression in leaves, for example, in several studies. And when you look at that, you see that about 1/3 of all the genes that are expressed in the leaves go up during the day, because the metabolism, photosynthesis is now occurring, and the metabolism is very active, and during the night goes down again. 1/3 of the genes are regulated every day. Wow, that's really as if the plants, well, are always optimized to their conditions that they need, light and night, but as if they still need to be responsive to this new condition light, new condition darkness. Okay. So another kind of memory or priming. Priming is very often used sometimes for abiotic stress priming, sometimes for priming for systemic acquired resistance for biotic stresses. That you have exposure of the plants to a certain stress, or to biotic, whatever it is, to a disease. And the plant responds to this, and then can respond at a higher level. Yeah? With biotic responses, we know that. Systemic acquired resistance is one of these features that occurs there. During vegetative growth, repeated life cycle, repeated stress response, yes, There are even some molecular players known, for example, for transient cold or heat stress for a few days. And then, well, the plants may remember that they have been exposed to the stress, and they don't want to go into much detail there. These are some epigenetic factors that work there. For transgenerational memory, which means memory in the next or in next generation, very problematic. So when I since a while read always these papers on potential transgenerational memory, and I agree with many other colleagues in the field. I would say not really well known, but probably not. We have evidence for the next generation, for flowering time, for salt tolerance, or osmotic stress, that there is a transgenerational memory, that the plants, even the seedlings derived from seeds from this exposed generation, these seedlings are a little bit stress tolerant for the next generation. But yeah, not more, not in the second generation. Nutrient memory or influence of nutritional status on memory, very little known, probably also not much at least. I will show you a little bit data that might go into that direction, that there might be something. Okay, so with this introduction, I want to ask you whether you have some examples, and I discussed already with one colleague here a little bit. She told me about a perennial plant where we don't need this transgenerational memory to be in many seasons, which I find interesting, especially in trees. I think this kind of memory is extremely interesting. Trees that live in a site, well, your oil pumps, you said, for 30 years. I would say our old trees in our forests in Germany for 600 years. Do we have these adaptations? Do we have this kind of memory?
Yes. What I was sharing was that little palms in the nursery that had been rejected for being small or crooked, not the right size, are sometimes helped. They go to the emergency room and take extra care. And when they have the right size, then they are planted. And that effort in a long time does not necessarily end up in a high-yielding plant. Because it, from the beginning, was not good. So even though we try to make it better on the long time, we cannot have a high-yielding plant. So I don't know how much is that from memory.
Yeah, so I repeat for I can speak up a little louder. So these plantlets that you had, they were a little smaller, discarded initially, and then taken care of a lot of, and these still did not keep up with the best plants that you had, as if they memorized some bad times. So plants sometimes seem to memorize some bad times. That's what we also find relatively frequent in the literature, that plants very often, or crops, somehow better remember bad times, and when they were exposed initially to some bad conditions, they are a little bit cautious to grow too fast and save their nutrients, and this is more typical. That's what we typically find in the literature. That's what I think is kind of memory, but it's only the response to bad memory. It's to be careful to grow. Yeah? Very rarely, I would say never, I saw that the plants benefited from good memory. Yeah? So when breeders, for example, grow their seeds for farmers, these fields where the seeds for the real production later are produced, they are very well maintained. They are in the best conditions. If these fields for seed production are exposed to severe stresses, the farmers who get the seeds probably will complain that the seeds do not really perform so well this time. So that's normally what the breeders do. They fertilize maximum, and everything is perfect, so the genetic material more or less remembers good times. Other examples that somebody has from you of kind of memory of plants? Maybe good memory. I'm always interested in good memory. No? Well?
Something that I heard from a biologist once was with a plant that is attacked by ants.
Yeah.
And so for, let's say, the next bloom, the next, when the shoots
Okay.
are going again, so that plant can be protected.
Okay, it would be go in the way of systemic acquired resistance against a biotic attack. So maybe salicylic acid was used or some defense compound also. Yeah, for biotic stresses we have this kind of priming or systemic acquired resistance. Yeah. Also in the next generation, it was trees.
No, it was not trees.
Okay.
It was, what she told me was the next generation.
The next generation, okay. Okay, that would be transgeneration. That would be really interesting. So I think there are really few examples. Still, the thing why many people think this is still something we should explore is because nowadays we know that in principle the epigenetic machinery for such kind of memory is available to the plant. So in principle, the plant might have the machinery to memorize something, and this I want to share with you today. So, and this machinery is called this kind of epigenetic machinery. And before we come to epigenetics, we have again to look at genetics. So genetics basically is the science where basically the phenotypes of genes are looked at, and where genes and phenotypes are inherited, and well, one gene, one phenotype, or the number of genes, a mixture of phenotypes. I told you about quantitative genetics, and now this genetics only works basically with the basis of DNA, with sequence information, and epigenetics is now on top of this genetics. On top of the sequences, we can apparently inherit something more. And the mechanisms behind that, I have it summarized here. For some plant like Arabidopsis, we have, of course, different tissues in such a plant. And this is, well, for a naive person, it's quite surprising because every organ here, every cell, has the same genomic DNA. So the same sequence is basically available, but only part of the sequence is used, and then a phenotype, a developmental phenotype, is somehow made from these few genes. And somehow the plant has to decide in some feedback mechanistic loops at which developmental stage, which genes are used, and which phenotype this later is. And this seems to be a relatively robust network and feedback loops that work there because normally all flowers look more or less the same. All leaves look more or less the same. Rarely problems with development of these. So all have the same genomic DNA, but apparently not all genes are always read and expressed in these different types of organs and cells. An old definition of epigenetics, one of the first by Weddington was, you have always the same genetic information, but it's used differentially in developmentally distinct tissues. So on top of the genetics, on top of the genes, you have some genes that are read, some genes that are not read, and expressed and non-expressed. Okay. So, today we have two major definitions of epigenetics, which epi simply means in Greek on top of genetics. It's the same DNA sequence, but on top of this DNA sequence, something is there. And epigenetics can investigate inherited changes in gene expression that do not involve changes in DNA sequence. So some information in addition to the DNA sequence information must come there. And for inheritance of this information, the following experiment could be done. You start with genetically uniform start material, genetically uniform seeds, and put them to different environments separately, half to high temperature, half to cold temperature. And then you put them into a common environment and look at the next generations. Or you look at the same generation, sometimes maybe for perennial, that's easier, for a tree or something. And well, for transgenerational inheritance, you would look in the F1, F2, and so on generation. You would self these plants and look whether they remember that they had been in the cold environment and whether they had been in the warm environment, are bigger or smaller. And we can look at gene expression, what is really different in these cases. So it's not really what we expected from an adaptive plant and simple genetics. That must require some additional information that is inherited. And this kind of Lamarckian type of inheritance now gets more possible, I would say, with the increasing knowledge about epigenetic mechanisms that we identify in animals and in plants. Never unambiguously shown for more than one generation, I would say. But, well, when I went to school, the biology school books, they mentioned that there was, at a time when Darwin was suggesting his theory of evolution, there was at the same time also Lamarck and Lamarckists that said, oh, we think that some acquired properties can be inherited. And in school books, it was made really clear Lamarck was completely wrong. Well, when you read really what Lamarck suggested, in many things he was really wrong, but the general idea of inheritance of acquired properties, this gets possible again. And as we as school pupils laughed about Lamarck, how stupid he was, Darwin was so clever, he was right. We tend to think that there may be, in some cases, even some Lamarckian type of inheritance. Okay. So the other biochemical definition of epigenetics is a change in chromatin. And what is chromatin? Chromatin is this assembly of genomic DNA with proteins, and I just mentioned yesterday, these histone octamer complexes with DNA is chromatin and some additional proteins. And biochemical definition of epigenetics is then it examines biochemical changes of chromatin, permanent, almost permanent, non-permanent changes in chromatin, regardless of the evidence of inheritance. That's a little bit weird because we talk about genetics, and with genetics we think about inheritance, but this is only biochemical way. These two levels of epigenetics, they somehow fit to each other relatedly, because the proteins or genes involved are more or less the same. So, examine the processes that are attached to DNA, stable or heritable, or more or less stable changes in gene expression, without changes in DNA sequence. And important keywords here, are these modifications of histones. Yesterday I mentioned acetylation, adding an acetyl group to the N-terminus of the histones, or adding methyl groups, 1, 2, 3 methyl groups to the N-termini of the histones. But also phosphorylation has been found, ubiquitination, adding a small protein called ubiquitin, a small ubiquitin modifier, ZUMO, can be added, or it can be ADP ribosylic. All these changes have been suggested and found, most important, acetylation and triple methylation. Yeah, chromatin modeling is one of the keywords that we have to use here, exchange of histone variants and change in DNA methylation in different contexts. We discussed this also briefly yesterday. And then we have the third thing, that is small inhibitory or small interfering RNAs of 21, 22, or 24 nucleotide lengths. And these can also be inherited independent of the chromatin and of the genomic DNA to filial cells. And they can have roles on setting DNA methylation cells and interfering with RNAs and degrading RNAs. So, this picture I showed more or less yesterday. It shows the histones and the DNA wrapped on these histone octamers, the modifications here on the histones, and the methylation on the DNA. And yesterday we discussed a little bit that apparently during, what was it, zinc deficiency, we had a big loss of these methyl groups on DNA, or genomic DNA. And from this, after 3 weeks of zinc deficiency, we had these losses of DNA. And we looked whether these losses of DNA was then correlated to some changed gene expression. And there was a mild correlation, not too strong, it was a little bit, with loss of this methylation on this DNA and gene expression. So some genes that were now expressed under zinc deficiency had lost this methylation that was found. Okay, and on the other hand, we discussed also in certain contexts how methylation is found in the CG context, this CHG and CHH context. And I said, especially for the asymmetrical context, CHH, we need some small RNAs that have sequence information that can somehow bind here to this genomic DNA and set methylation at certain positions. A more detailed scheme of this can be found by some reviews. I don't want to go into the details there. What I want to show you is that even though we have analyzed now many tissues and many plants for these kind of chromatin modifications and compare this to gene expression, we don't have a really very good picture what these modifications do. So when we have a gene like this here, this is the gene that has a promoter here, 1 kilobase promoter in front. Then we have the transcriptional start site. We have a 5' untranslated region of the gene. Then we have the transcribed and later translated gene to a protein. So this is non-translated. Here is also a 3' at the end, 3' non-translated region. All this here is exon, so it's translated into protein later. And then we have a short sequence behind the gene somehow. And we look whether this correlates with transcription when we have a nucleosome formed there, which means there are histones found attached to this sequence. And whether we have methylation in the CG context, for example. And it seems when we have methylation in the DNA, well, when we have methylation here in this transcriptional start site, it seems to be negatively affecting transcription. When we have methylation in the gene body, well, Well, it seems sometimes positively correlated. It's not so clear, positive or negative. When we have histone 3, we have 4 different, well, yeah, 5 different histones. Histone 1, histone 2A, 2B, 3, and 4. And mostly these histones number 3 are those that are methylated or acetylated or modified in their N-terminus. They're most different. When we have this lysine at position 4 in this histone number 3, methylated ones, well, no, not really known positive or negative. It can be everything. What is informative? Informative is this here, 3 times methylated, the lysine in position 4 in histone 3. When it's methylated here at the beginning of the gene, typically this is associated with gene expression, positively correlated with gene expression. Then we can go a little bit further here down. This is negatively correlated with gene expression. Lysine 27, 3 times methylated at histone 3. When it's positioned somewhere in the gene, well, along the entire gene, it's negatively correlated with gene expression. And then we have here an acetylated lysine number 9 at histone 3, or acetylated lysine at position 27, also methylated, positively correlated. So acetylation is typically positively correlated with gene expression. So all the others, well, we don't know really what they really do. So when we look at methylation of DNA, not methylation of histones, only methylation of DNA, then 2 species have been investigated most in detail. This is Arabidopsis and maize. And for Arabidopsis and maize, we have a certain amount of methylation in the genome. And most important are, well, we have this CG methylation, we have CH demethylation and CHH methylation. And here, well, the comparison is shown what is typically found in the genomes of Arabidopsis and maize. Arabidopsis is a genome which is very compact, relatively few transposons, many genes very close to each other. Maize is the opposite. Genes very spread over a long region, many transposons, many regions between the genes, maybe regulatory regions. The majority in Arabidopsis is this here, this kind of green color, which is unmethylated. So the majority in Arabidopsis is unmethylated. And in maize, the majority is CG, CHG only methylated. So the majority in Arabidopsis unmethylated, in maize, the majority is CG or CHG, so symmetrically methylated. What does it mean? Well, as I said, in maize we have a lot of transposons. Transposons are silenced by this kind of methylation. In Arabidopsis we have very few transposons. We don't need to silence. So genome organization is quite distinct. When we now look at the different kinds of methylation in Arabidopsis, we see that most of the methylation here is close to the centromere in one chromosome. This is one of the 5 chromosomes. This is one of the 10 chromosomes of maize. But all chromosomes are quite similar to each other. So unmethylated, most of the part of the chromosome in Arabidopsis is unmethylated here. Only in the centromere region, where also some transposable elements are. There are also transposable elements here, but not so many. It's unmethylated. For maize, it's completely different. Most of the chromosome here is symmetrically methylated, at least in part. Very different importance of this methylation probably in these 2 species, in Arabidopsis and maize. For maize, unfortunately, we know much less. about the enzymes that are involved in maintaining this methylation, in setting new methylation, or removing methylation. For Arabidopsis, we know much better which enzymes are there, and we try to deduce from the enzymes that we find in Arabidopsis how they work in maize. For Arabidopsis, we have a lot of mutants where certain genes for maintenance of DNA methylation are knocked out. And what is the methylation pattern that we typically have? The methylation pattern that we typically have is in front, in the promoter of the gene, we have some kind of methylation that goes down at the very transcriptional start site, goes up in the gene body again, and is low at the end of the gene again, and then goes up again. This is the normal CG methylation. CHG methylation, CHH is slightly different. This is here the most prominent methylation in Arabidopsis. And when you knock out one enzyme that is, or one gene that is called MET1 for methylating methyltransferase number 1, when you knock that out, this CG methylation is gone. CHG methylation, CHH methylation is a little bit changed, but not much. And the interesting thing now is, how do the plants look? Do they survive when MET1 is knocked out? And the answer is, they look pretty normal, surprisingly. They have higher mutational rates, and next generations will look a little bit more funny. Quite well. But the first generation looks pretty normal. Yeah, you have other genes and enzymes that are important for maintaining some of the methylation here, and some mutants I had shown you with respect to zinc. Effects on zinc deficiency. One mutant that was this here, that was the so-called DDC mutant that I had in one slide, that looked funny on a zinc deficiency, that was this bushy phenotype, on a zinc deficiency, not nice flower development, and it has lost most of this non-symmetrical or CHG methylation, non-CG methylation. Okay, so this is the background to this DNA methylation. What we have in Arabidopsis, we know most. In maize, we also have some mutants where methylation is slightly reduced, but not so globally and so strong in these different contexts. So, what have we learned about DNA methylation from others recently? So, in Arabidopsis, researchers finished to sequence 1,000 or so genomes. They still had the sequencing machineries and still had money left over, and said, why not sequence all the methylation patterns in all these 1,000 genomes that we have already sequenced now? And with some techniques called so-called bisulfite sequencing technique, they can measure which cytosines are methylated and what the methylation phenotype looks like. And they sequenced 1,000 genomes from Arabidopsis, from mostly European, Asian accessions from Arabidopsis. And what they found is that genotypes growing in warmer conditions typically had less methylation than genotypes growing in Sweden or colder environments in the north. Surprisingly, this was one of the few things that really came out of this sequencing of so many genotypes. Not so much informative information what this methylation really does. Mostly what came out was more methylation in environments where genotypes are exposed to colder temperatures. One study that I found extremely interesting, and we try to repeat things that they did in maize, was in Arabidopsis, was done mostly in rice, but also they added some Arabidopsis data there. So what did they do? They exposed rice plants to P deficiency in hydroponic conditions, so nutrient solutions, and they analyzed the gene expression after exposure to P deficiency, after a short time, after longer times, put plants back to normal nutrition, and at these time points always also measured the methylation level in the genome of rice. And what they found was that under normal conditions with phosphorus, certain phosphorus starvation-induced genes were not expressed because they are only under starvation expressed. When you are now on a short-term starvation or long-term starvation, apparently these genes got expressed, but somehow these genes got expressed and transposons close to this gene were somehow, or transposable elements close to this gene, were high pair methylated. Not always, but sometimes. So apparently, as if the expression of a gene under P deficiency stimulated the methylation of a transposable element close to this gene. Yeah, this is very, very interesting because it may mean that methylation then is important. And this methylation of a transposable element here may have a secondary effect on the gene expression of this gene later. We looked at these kind of experiments or similar things, not so much in detail because we did not have so much sequencing money for it, on nitrogen, phosphorus, and zinc deficiency. Some zinc deficiency results I showed you yesterday. With nitrogen and zinc deficiency, we find a lot of reduced methylation. Half of the methylation or so is eliminated. With P deficiency, we did not find this, and it was not very well correlated with gene change. So these are some results of the methylation levels across the chromosome 1. Okay, so probably this effect in rice with DNA methylation and P deficiency may be specific to rice, not found in Arabidopsis, not found in maize. But probably DNA methylation close to genes is important because there's one study that shows directly biochemically that motifs for transcription factor binding can be methylated or cannot be methylated, and the change of the methylation changes the affinity of the transcription factors to bind there. So genes that are under control of certain transcription factors, their expression depends on whether a transcription factor can bind there, whether this motif is methylated or not. This is very much affected by methylation. It's a lot of transcription factors that are involved in stress signaling that have been shown to be regulated by methylation, and methylation in most transcription factor suppresses transcription factor binding. There are, however, of the many, I think 600 or so, they investigated of transcription factors, some even are stimulated by methylation, transcription factor binding. I would like, at the very end, share with you some experiments that we did along these lines with a perennial plant, a little bit, I would say, similar to your oil plants, with poplar, which in Germany is grown for biomass production in small rotation farming areas. And there, what we do is we often use stem cuttings for establishment of these, which means that there is genetically uniform material that is used, often at different sites in Germany, also at different P levels, these sites are. And we got access to some of these sites and analyze this a little bit in some molecular detail. Actually, the different sites that we were looking here it's basically this kind of epigenetic experiment that we discussed a little bit earlier. We have genetically uniform material. We have two kinds of conditions. We have stress conditions with less phosphorus in the soil and optimal supply of phosphorus in the soil. But in addition to the phosphorus difference, we also have completely different environments. So both sites where we collected the wood were in northern Germany, but both were exposed to completely different weather. Well, not completely different weather, but different rain times and so on, different amounts. And then we put them into a common environment in our greenhouse, in our growth chambers in my laboratory. And we looked again at plus P and minus P and whether we find a phosphorus-related effect, because this previous research in rice suggested that there may be some kind of methylation memory effect in rice. So unfortunately, yes, as I said, it's not only that it's two sites with different P level, it's also two sites, one was called Underlingen, one was called Waldstaden, the one is here next to Bremen here, and the other one is about 60 kilometers or so away from this in Waldstaden. These were two sites. Underlingen was exposed to more stress, apparently, and Waldstaden was better supplied with phosphorus. Here are our P analysis from the leaves, wood, soil from these two sites. We had several other sites which we later discarded. We only focused on these two sites. And what do I see here? Underlingen and Waldstaden. This is P concentration of the leaves. We got some leaves from trees in these areas, and in Waldstaden the P supply was much better in the leaves. In Underlingen they had P deficiency, in Waldstaden not. And this was also related to the P concentration in the wood from the stem cuttings that we got from these sites, and the P availability in the soil that we analyzed, which was, well, here only half of the other. Okay, so let's start with this material and analyze it in the laboratory. When the stem cuttings started to develop, they make some roots, very nice roots, under P sufficiency and P deficiency. We looked at this. And we looked at the first leaves. We looked within, I would say, 10 weeks or so. Later, typically these poplar trees in our conditions got some problems in nutrient solutions, and we looked only at relatively early events. But anyhow, these Waldstaden, well supplied with P, they somehow seem to develop a little bit more robust, and we have the tendency to also see, I can see more roots. And here, this here is a little bit of anthocyanin color coloration. It looks a little bit maybe P deficient and stress exposed somehow. Okay, when we look at this in a little bit more quantitative way, we could get from this well-supplied stems, we could get more dryness. So the seedlings that established from the stem cuttings, they were establishing much better, both under plus P and under minus P. And this other stem cuttings from Underlingen, which were genetically uniform, same material, they were establishing worse. So somehow, yes, they remembered where they were grown. The stem cuttings looked very similar or identical. Dry root biomass was, total biomass, was always bigger with this well-supplied clones. What went in here? The P concentration, well, this was not different. Total root length, yes, the Waldstaden had more roots. It rooted better, it grew better, it was somehow establishing better. And we said, well, there is some memory, let's look at the epigenetic state, let's look at DNA methylation. And we did it in Waldstaff and Underling, and we had some regions where apparently there was less methylation in some certain regions. There was not so many regions that were differentially methylated, but there were some. And according to our criteria, we found that there were, well, what was it in total? About 200 regions that were differentially methylated, some more in this one genotype, one more in the other genotype. Some in the gene body, the promoter, or overlap. And because we knew that, especially at the beginning of the gene, at the end of the promoter, there typically DNA methylation is negative, correlate with gene expression. We looked at most of these genes that were methylated in this region. And we looked at the 40 top differentially methylated genes, whether they were differentially expressed. And unfortunately, of the top differentially methylated genes, only 6 were expressed at all in the roots, all the others were not expressed. So for all the others we cannot say anything, but for these 6 that were expressed, in the 6 meter plants, we get this picture, and we get the picture that in underling it was always some kind of standard expression, and in Waldstaff it was more variable somehow, the expression. Both in leaves on a plus P and a minus P condition, the phosphorus concentration did not change so much of the gene expression. In roots, again, the underling was always more or less, the genes were standard expressed at some level. The Waldstaff were more different somehow. Again, both under plus and minus P, the P level did not affect as much. So there was not an indication that this differential methylation was really having an effect on the gene expression. And we were quite disappointed, and then realized, this PhD student who did that, she realized that there were some microRNAs, which are small transcribed sequences that not encode a full gene, but encode pieces of genes that are complementary to genes. And these can be processed, these microRNAs, for secondary regulation and downregulation of messenger RNA expression. And some of these microRNAs, they were close or hit by our differential methylation in these 2 genotypes. It's also a cloud of points here for these few differentially methylated regions where we had microRNAs in. Overall, well, it's not really very consistent, but again, somehow this underlingen was quite constant somehow, normalized gene expression, and the differential methylation in roots was for Waldstaff, well, a little bit more, not so constant at least. When we look at the entire messenger, the microRNA to small RNA ratio in total in percent, we see quite some differences between the different sites. Site-dependent in leaves and P-dependent in roots. What this really means, we do not really know, but we think, well, it's kind of memory that may be there, that might be investigated also in the future, and maybe in oil palms. We finished with this work with poplar because we realized that really poplar is a mess to work with. We have a fully annotated genome, but still compared to Arabidopsis, it's still a nightmare. It's still not very well annotated. And with Arabidopsis, it's simply easier to work with. Okay. So, conclusions from my lecture on yield stability and memory. Yield stability is a quantitative trait. Physiological knowledge may help for specialized genotype for specific environments, but not so much in general for quantitative traits. Flowering is a key trait, definitely for broad tolerance, especially terminal drop. Epigenetics mechanism provide a mechanism framework to understand memory in priming implants, but still there's relatively little evidence for that, especially for transgenerational memory, not more for more than one generation. Yeah, there's some kind of memory in poplar stem cuttings. They memorize somehow nutrition and biological history. It may, of course, be something different than this epigenetic mechanisms that underlie this memory. It can be simply a metabolite that is found in one stem cutting and not the others. This has not been yet sufficiently investigated, but it is probably relevant for the local adaptation and planting of these stem cuttings for trees. Good. With this, I'm finished. So I had a lot of fun in this lecture. I'm very happy that many of you asked questions, and if there are more questions, I'm happy to answer them. And if not, I'm finished for this week. Thank you for your attention.